I would like to share with you the right part of the picture, because what the reviewers in Nature Genetics says that this is right now a sort of poster model of how people should be dealing with this kind of a study. So first of all, there were some 7 million polymorphisms done in genome-wide association study, including 800 cases and 37,000+ controls. And you might say, hey, I just said that there are up to 4 million polymorphisms and here there are 7 million polymorphisms. That's true because there are methods to so-called imputation of polymorphisms. So you can actually show the results for polymorphisms that you have not originally genotyped. Of course, if you find an association, it's obvious that you have to genotype this polymorphism later, but you don't have to do it up front. And in this group of patients, the association with the polymorphism was found. The polymorphism was frequent. However, what the authors of the paper did as a next step and a third step over here, they did whole genome sequencing of just 87 people. So we have to remember that in such studies, we don't really have to genotype to sequence 30,000 people. This would be way impossible. But you can just select a group of people. They have found a missense mutation in a gene which is cardiac myosin, so a perfect candidate gene for such a sick sinus syndrome. Then the direct genotyping of this polymorphism and found that this was very highly associated with the sick sinus syndrome. So another proof of evidence was here that people who were healthy but did have this polymorphism, because as you remember, only 50% of people will develop sick sinus disease. But the other half of the people, they were healthy, but they clearly had a slower heart rate and they had a larger PR interval in their ECG electrocardiograph picture. So this underscores the biological importance of this polymorphism. The other potential explanations for missing heritability, the structural variants, the copy number variants, and gene-gene interactions. So we know at least that copy number variations have been already implicated in several complex diseases, including diabetes, including myocardial infarction, for example. There is an ongoing debate, and I do not have own data to sort of take a stand here, because some researchers claim that current microarrays adequately capture copy number variation, so we will not learn more from doing specifically copy number variations studies. The others would strongly oppose to that, and they would say that current microarrays have relatively poor capability of detecting copy number variations, and we need better techniques, and we need to specifically address this question. Next generation sequencing, at least some of these methods, can help with detection of structural variants. With gene-gene interactions, the hypothesis is that it's not the gene itself, the polymorphism in the gene, that is causing the disease, but the interaction with the other polymorphism in a completely different gene on a different chromosome, for example. So there are several problems here. First of all, in general, to detect such effects, you need much larger sample sizes. The case-only specific type of studies can help, but not really much. These studies are computationally intensive. If you compare 4 million polymorphisms, each of these 4 million polymorphisms in your microarray with another 4 million polymorphisms, you do it pairwise, it takes days, sometimes weeks, on pretty strong computers. If you would like to study higher order interactions like 3 genes, 4 genes, this becomes hardly manageable even for large sequencing centers. And this is puzzling a little bit because, at least theoretically, there is a possibility that you statistically see a gene-gene interaction without seeing gene effect itself. Because the easy strategy would be to select first significant genes, significant polymorphisms, and test just pairwise all that is significant in the initial screen. This is a good strategy, but we have to remember that it will not capture all the possibilities. And finally, I would like to briefly touch upon a paper which was published last week in Nature Genetics about the authors who very strongly suggest that there is no such thing as missing heritability. What they show here are 4 different traits. A is height, B is BMI, C is von Willebrand factor, and D is, I believe, QT interval, so one of the electrocardiogram features. And what they see very clearly is that there is a very strong correlation between the length of the chromosome and the variance explained by each chromosome. So if you have a large chromosome, like chromosome 1, 2, 3, you will see more variance explained than by small chromosomes like 20, 21, 22. And this is in opposition to what you would expect. You would rather expect that some chromosomes would actually harbor the variants of interest, the polymorphisms of interest. They would sort of stand out of this line very significantly because they would harbor the polymorphism, the culprit polymorphism. This doesn't seem to be the case. So what they say, according to their method called genome partitioning, on the right-hand box you see these 4 traits and their heritability based on various previous studies. The heritability is quite high, 80 to 90% for height, 30 to 60% for QT interval. So rather strong heritability. And in the blue box, you see the heritability that is currently explained by genome-wide association studies that have been performed so far. And you see it's very low. So the rest is missing heritability. That's what I have talked about. However, these authors claim that according to their method, they can explain much higher heritability, much higher than previously thought. And they believe that this is due to association with polymorphisms which did not find genome-wide significance, which were not of genome-wide significance in previous genome-wide association studies. It is true that if you look at these plots, you find a couple of polymorphisms that clearly stand out above the genome-wide p-value, but there are also the areas with lower p-values. And what the authors propose in their paper is that actually we have to address this in one of 2 ways. Either increase the size, genotype it, about 100,000 people, that's what they propose, and also look for variants that are less common and not tagged with current methods. So at the end of the day, I'm happy to say that genetics comes back to old good days when everybody was saying that we need to genotype more people and we need to genotype more polymorphisms. So much has changed in order that nothing has changed, in a sense. And in the final slide, the conclusions that I would like to say is that genome-wide association studies solely reach the limits of their usefulness. Because you can always add more polymorphisms, but these chips will be more expensive, and with the dropping cost of sequencing, it will always be better to know 3 billion answers, 1 answer per each letter in our genetic code, than to have 4 million, 5 million, 10 million answers for pretty much the same cost. So 3 billion will be better than 10 million. Next generation sequencing certainly offers promise for the traits with an underlying genetic model of rare variants with very strong effects. However, there are clearly situations in which, like gene-gene interactions, when next generation sequencing will not be very helpful. So there are clearly situations when that this is not a uniform solution for every kind of genetic study in the future. And finally, many researchers say that although it never hurts to have more samples and more genotype polymorphisms, at the end of the day we will not avoid carefully designed prospective studies in which we can truly dissect environmental factors from genetic factors, because there are possible biases in the retrospective case control studies that are very hard to control overall. Thank you very much.
Thank you. We have time for a couple questions. Alessandro?
Pavel, that was really great overview of where we are and where we are going. Now, just a very simple question. All those talks about a missing heritability, they assume that we have the right measure of heritability, right? And so we go back to the, what do you think? Do you think that there's any inflation of heritability due to familial clustering of other factors, or you think that we have a fairly reliable estimates? And then I have actually a second thing that might actually go back to your background as a quantitative person. When we say that the rare variants have a larger effect, now this is under the framework of a risk ratio. But if you look at risk difference, actually, basically they have the same effect than the common one. So I'd like you to comment on this as well.
Okay, so I remember Alessandro from Boston asking questions that seem to be simple, but they are very, very, very insightful questions. So I mean, I can only give like a sort of my best guess, and I think the estimates of heritability are probably a little bit inflated. So we will see a decrease over here. You see also that there is a large variability, like from 30 to 60%. So these measures are not stable. And it's also well known that heritability changes, heritability in a population changes depending on what kind of environmental factors are present in the given populations. So if you study a pretty uniform population with pretty uniform environmental factors, like for example, only people living in rural conditions or only people living in the city or so on, then heritability will be larger because the environmental factors are diminished here. So I think what we see when we see that heritability is from 30 to 60% is just pulling much variable studies, and that's the difference. And your second question was about risk ratio and risk difference. I mean, yeah, I cannot agree more. I mean, I would definitely like to see the prospective studies in which you would truly see the risk rather than odds, because we know that the odds ratios are interpreted under certain circumstances, and we don't know whether they are fully fulfilled.
Okay, I think in light of the time, we'll move on. Thanks again, Pavel.